Okay, so
Okay, great. Welcome to today's Immune Zoom seminar. It's my absolute pleasure to introduce our speaker, Dr. Chris Thaiss, who is currently an assistant professor at the Department of Microbiology at Penn Medical School. Chris performed his undergraduate studies in molecular biomedicine at the University of Bonn, Germany, and his master in microbiology and immunology at Yale University and ETH Zurich, Switzerland. After a short-term scholarship at the Broad Institute of MIT and Harvard, he performed his graduate study at the Weizmann Institute of Science in Israel, which also include a visiting fellowship at Stanford University. So as you can tell, Chris is really well-traveled during his entire training period and gets connected to, I think, all the different cultures and also, I think, research entities. After completion of his graduate work, Chris established his own research group at the University of Pennsylvania. Currently, his lab studies the role of body-brain interactions in metabolic and inflammatory diseases, with a particular focus on the role of intestinal microbiota in the regulation of host physiology. His groups exploit system biology tools, including single-cell transcriptomics, metagenomics, and metabolomics, coupled to hypothesis-driven research in animal models and human cohorts. The ultimate goal of his lab is to develop and innovate new therapies for common human diseases. So without further ado, Chris, the floor is yours.
Great, thanks very much, Ole. Thanks for the invitation, and I'm really excited to be here and give this talk to all of you. I have one small disclaimer before I start, which is that I'm alone at home with our two young kids who are sleeping. I hope they will continue sleeping because if not, then they will probably join me here in this talk, which will be fun for everybody. So let's get started and I'll hope for the best. But basically the theme of my group in general and everything that we strive to accomplish has to do with what you can see here on this slide, which is showing us some of the most common and major human diseases, including neurodegeneration, cancer, metabolic disease, and chronic inflammatory disease. And our goal is to identify commonalities among all of these diseases, not only because this will allow us to come up with new treatment and prevention options that will act much earlier compared to what we have available right now, but also because biologically it's extremely interesting to try and understand these commonalities from an evolutionary perspective. Because in many cases, we don't have a very clear understanding why these diseases exist and what the physiological counterpart might be that was selected for during human evolution. So right now, we basically have two major theories why these diseases exist and what the commonalities are between these diseases. And I'm sure you will all know about these two theories. The first one is that these are diseases of aging, right? That the older we get, the more susceptible we are to these diseases because they're basically byproducts of waste accumulation in a way that happens over the lifespan, and then these lead to susceptibility to these diseases. And if that's the case, then we would postulate that selection has not occurred because these are largely diseases of the post-reproductive period. But there is at least a second major theory, which is the environmental mismatch hypothesis, which says that selection has occurred and there are physiological counterparts, but they have been selected for during a period of human evolutionary history that was completely unlike our current environment. And so now the food we eat and the things we do in our daily lives predisposes us to some of these very common human diseases. And my lab is studying the intersection of these two theories. And the way this looks like in the lab is we view a human being as the result of all of these different environmental exposures, everything we do in our daily lives, from basically what we eat, how much we sleep, how much we exercise, how stressed we are, and so on, including time as an abiotic risk factor, which is basically that leads to aging and predisposes us to disease as well. And the way we approach this in our experiments is by viewing these environmental factors through the lens of what we think are the three most important sensory entities of the human body, which are the immune system, sensory epithelial cells, and the microbiome. And then we're specifically interested in understanding how all these sensory events are integrated by the nervous system, and how at the level of the nervous system, our susceptibility for disease is then regulated. And so for the purpose of today, I thought I'll show you a few examples of how we have studied this recently in a few specific projects. And for the purpose of this audience, I decided to focus primarily on those pathways and environmental factors which we think involve a strong inflammatory component. And the first one I'll talk about is one of the most detrimental environmental factors here on this slide. And what I'm talking about is psychological stress, because we know that psychological stress from many, many decades of epidemiological studies, we know that psychological stress is a huge risk factor for the exacerbation of peripheral inflammatory diseases. And so this is something that was first initiated in the lab by a trio that you can see here. Markus Schneider was a postdoc in the lab who now has his own lab. Nicholas Blank is a PhD student in the lab, and Yelena Alvarez was a gastroenterology fellow in the lab who is now working with Dan Musida at the Rockefeller. And what Yelena brought up to me early on was that from her own experience, as well as from the medical literature, there's a lot of associations, for example, in this example here, between self-evaluated psychological stress of a patient and the proportion in remission. In this case, these are IBD patients. So these are individuals with inflammatory bowel disease, and you can see here that the higher the psychological stress, the worse the long-term outcome for these individuals. And of course, that's just one out of many, many such studies. But even though we have this association in very strong clinical literature, there are actually very few mechanisms known by which stress perception in the brain leads to the exacerbation of a peripheral inflammatory response, for example, in the case of IBD. So we thought, how can we study this in a mechanistic way? And we can actually study this in animal systems because there are many different ways to recapitulate psychological stress in an animal. Among the most common models are the restrained stress system and the social defeat stress model. And then we can pair those with different models of IBD. For example, DSS is one of the common ones. We can use TNBS, which is another chemical, T-cell transfers, IL-10 knockouts, and many other IBD models that we have. But whichever combination of stress model and IBD model we used, we always saw the same phenotype, which is strongly exacerbated inflammatory disease, which you can see here by a few of the readouts that we use, such as weight loss, colonoscopy evaluation, and then an aggregated score. So we can definitely recapitulate the connection between psychological stress and inflammatory bowel disease in animals. But now the question is, how does this work? What's the mechanism downstream of stress perception? And because there were so many options, we resorted to single-cell RNA sequencing. So we looked at colonic immune cells in stressed mice and control mice. And I'm highlighting the myeloid cells here because the myeloid cells turned out to be very interesting. As you can see here on the right, if we use a pseudotime algorithm to line up the cells across a putative differentiation trajectory, we see what we expect, which is the differentiation from monocytes to macrophages, because in the GI tract, as you know, monocytes infiltrate from the GI tract, from the circulation, and then they differentiate it into tissue-resident macrophages. But what was really interesting here was that in the stressed animals, there seems to be an accumulation of these monocytes on the left side. So the differentiation trajectory is stuck on the left side, if you will. And we can verify this by flow cytometry. Those of you working in the GI tract will know that this is the famous monocyte waterfall, which goes from Ly6C positive monocytes up here to MHC positive macrophages down here. And with a bit of imagination, you can see a waterfall here going down. Now, the interesting thing is that in stressed animals, this waterfall is broken. As you can see here, it's blocked, and you get this accumulation of monocytes, which are inflammatory. And I should have mentioned on the previous slide that these monocytes tend to produce TNF. And TNF, of course, is a very well-known driver of inflammation in the context of inflammatory bowel disease. So this was interesting to us because now it suggested that the accumulation of these monocytes may be a driver of the disease. And that's something that we tested specifically by either ablating monocytes or by inhibiting TNF, and that's what you can see here. So here we're using two different approaches. This is a CCR2 DTR mouse, which allows us to ablate circulating monocytes. And on the other hand, we use a monoclonal antibody against TNF. And in each case, now you can see here, even though we stress the animal, they're no longer susceptible to this exacerbated disease. So either we ablate monocytes, we see no difference to the unstressed animal, or we block TNF and we get exactly the same result, suggesting that the most common downstream end of this pathway involves monocytes and TNF production and leads to exacerbated inflammation. But now the question, of course, is how does the signal from the brain that perceives a stressful situation, how does this signal reach the monocytes? In other words, what is this arrow here, and why is it so long? How many steps are there in between? So that's what we next tackled. And of course, every time we think about stress perception by the brain and communication of stress to the periphery, we think about two canonical pathways. On the one hand, we think about the adrenergic system, which leads to the release of norepinephrine in the periphery. And as we would expect, this is basically what we see when we stress the animals, very strongly exacerbated norepinephrine levels. And on the other hand, the second axis is the HPA axis, which leads to the production of glucocorticoids, such as corticosterone. And this is exactly what we see here as well. You can see as we put the animals through this model, you see strongly elevated levels of corticosterone. So now, which of the two pathways is the one that exacerbates colitis? We first address this one here, and for the sake of time, I will not show you the results, but we didn't see any evidence pharmacologically or genetically that the adrenergic system drives disease exacerbation in a stressed animal. But for the other system, we actually did find evidence, because it didn't matter at which stage of this pathway we came with a genetic or a pharmacological intervention, we always saw the same outcome, which is what you can see here. So these are mice, for example, that underwent adrenalectomy. So these mice are basically lacking this stage of the HPA axis. They don't have an adrenal gland. And so they produce, the production of glucocorticoids is strongly reduced. And now you can see that these mice look like the anti-TNF mice. So they, even though we stress these animals, they don't respond with a stronger colitis. And the same was observed when we used mifepristone, which is a glucocorticoid receptor inhibitor. And conversely, if we stimulate the pathway, for example, with the glucocorticoid receptor agonists dexamethasone, now we get exacerbated disease, which was very surprising because steroids are, of course, first-line treatments in IBD. And if we acutely treat mice with glucocorticoids, we see the expected anti-inflammatory response. But if we give it chronically, then it seems like we turn this into a more pro-inflammatory outcome, which is very similar to the stress situation. So based on all of these results, we concluded that the HPA axis is basically the mediator between what's happening in the brain, most upstream, and the downstream monocytes and TNF exacerbation of inflammation. So at this stage, we thought, okay, now it's going to be relatively straightforward to understand, because the monocytes are known to express high levels of the glucocorticoid receptor. So we thought we'll do one more experiment to address whether glucocorticoids directly activate monocytes to produce TNF. And so we generated conditional knockout mice, which lack the glucocorticoid receptor NR3C1 in myeloid cells under the control of the lysozyme promoter. But now you can see here that this does not rescue at all the animals. They still respond with a lot stronger colitis if we stress them. So we tried other Cree mice, and the Cree mouse that gave us a rescue is HAN2 Cree, because as you can see here, if we eliminate NR3C1, which is the glucocorticoid receptor, on HAN2 expressing cells, now there is no longer exacerbated disease when we stress the animal. Now HAN2 was very interesting because HAN2 in the gastrointestinal tract marks the cells of the enteric nervous system. So as you know, there's a very dense nervous system that is local to the gastrointestinal tract, with both neurons and glial cells that reside in the wall of the gastrointestinal tract. And they're involved in many different functions of the GI tract, most prominently motility, but also secretion, immune regulation, many other aspects of GI function. And what's interesting is that HAN2 labels both enteric glial cells and enteric neurons. So we eliminated the glucocorticoid receptor in both of these populations, and we now had the challenge to identify whether it's neurons or glial cells that respond to glucocorticoids and then drive inflammation. So we used two more Cree lines, PLP1 and SOX10, which in the adult animal are specific to glia cells. And when we now eliminate NR3C1 specifically on glia cells, we get the same rescue from the exacerbated disease, which means that it's most likely the glia cells that need to respond to glucocorticoids in a stressed animal to drive exacerbated disease. So now the question is, what's up with these glia cells? So we again used single nucleus transcriptomics of this time of the enteric nervous system. We can categorize the cells by canonical markers into neurons and glia cells. And then when we zoom in onto the glia cells, we identify these 4 populations. The 4th one of which was very interesting because it only occurred in stressed animals, as well as in dexamethasone treated animals. So it seems like chronic stress as well as chronic glucocorticoid receptor signaling gives rise to these glia cells. And accordingly, we named them enteric glia associated with psychological stress, or EGAPS. And we know that glia cells had to be involved in the phenotype because of the following 2 experiments. When we ablate enteric glia cells, again, using either SOX10 or PLP1 as the driver with a diphtheria toxin system, now we protect the mice again from severe disease. So in the control, you can see if we stress the animal, they have more severe colitis, but not if we ablate these glia cells. And the same is true if we treat the mice with dexamethasone. So the glia cells seem to be a very important hub that mediate the effect of these glucocorticoids that are released in response to chronic stress, and the monocytes that then produce TNF. But we still have the open question of how the signal comes from these EGAPs, from these inflammatory enteric glia cells, and how it influences the biology of the monocyte. So that's what I want to discuss next. What's this question mark here, essentially? And here we were able to basically compare our 2 transcriptomics data sets, because you remember that we have these single cell transcriptomes from enteric immune cells during stress and during colitis, and we also had this single nucleus data set from the enteric nervous system. So we were able to use one of these interaction network algorithms that suggest possible factors of communication that might be released from the glia cells and then signal to the myeloid cells and vice versa. And one of these factors caught our attention, which is the one down here, CSF1, because as you know, CSF1 is a very well-studied monocyte chemoattractant factor that strongly influences the biology of myeloid cells. And there are also many suggestions of CSF1 being involved in the pathophysiology of IBD. So now the question was, is CSF1 the missing link between these glia cells, these inflammatory EGAPs, and the myeloid cells? So we did one more experiment in which we blocked CSF1 signaling using a monoclonal antibody. And now you can see that if we do this, the animals no longer accumulate these inflammatory monocytes in their GI tract, even though we stress them. So this is what you've seen before. These are monocytes accumulating in the GI tract of a stressed animal. But if we block CSF1, this is no longer the case. And if we do this, we can also rescue the mice from colitis exacerbation, as you can see here again, by weight loss and by colonoscopy, which is strongly reduced if we block CSF1 signaling. So based on this, I think we can fill another gap here, which is that CSF1 is probably one of the most important factors derived from these inflammatory glia that then signals to monocytes and leads to the production of more TNF and then more chronic inflammation in this setting. Okay, so that's basically what we were able to work out in animals. But now the question is any of these pathway active in humans in the context of IBD? So we addressed this using a cohort of 63 IBD patients that were enrolled in what's called the I3 study here at Penn Medicine. And what's nice about this study is that we were able to administer a questionnaire to these individuals. We were able to ask them many different questions about their lifestyle, and we were able to use one of the very well validated questionnaires that allows us to assess the stress of these individuals in their daily lives. So we have a stress score from each person, and then when they come in for a colonoscopy, we were able to obtain biopsies to look at clinical outcomes as well as a few molecular markers assessed by transcriptional analysis. And first, as we expected from the literature, as well as from a large-scale analysis in the UK Biobank that we did, we see a correlation between an individual's perceived stress score and their colonoscopy score. So we see more inflammatory parameters in these individuals with higher perceived stress. And then interestingly, we also see correlation with a few inflammatory markers, including those that we had studied in animals, including CCL2 and CCR2, which are important for monocyte recruitment, IL-6 and TNF and other myeloid-derived inflammatory cytokines. So at least preliminarily, we think that there might be a connection between psychological stress in human individuals and their inflammatory outcomes, which now motivates us to use this as more information potentially to inform clinical decision-making down the road. And that's something that we hope to pursue now in a much larger cohort of individuals. So with this, I think I'll switch gears and I will go to another environmental factor, which is certainly very detrimental and very important to anybody studying immunology or inflammation, which is infection. Because as you know, and of course, nobody needs a reminder about the SARS-CoV-2 pandemic, but infection in general, even beyond SARS-CoV-2, is of course a very important factor that regulates our health acutely. But now we're much more aware that it also regulates our health and predisposes us to disease in the long run. And of course, what I'm alluding to is this growing class of post-infection syndromes, which receives a lot of attention now in the context of SARS-CoV-2 and long COVID, but is by no means unique to this virus because it has been observed across other viruses as well as non-viral pathogens, many of which lead, in a subset of infected individuals, lead to these chronic post-infection syndromes. And these post-infection syndromes, they seem to affect almost the entire body. There are manifestations in many, many different organ systems. And what makes these conditions so hard to study is that we don't have a very good understanding at this point of the underlying mechanisms. There are several different suggestions of possible mechanisms that contribute to the pathogenesis of these diseases. For example, chronic dysregulation of the immune system, dysbiosis of the microbiome. There's evidence for autoimmunity that can contribute to some of the symptoms. There's also evidence for platelet dysfunction, blood clotting and endothelial abnormalities. And many of the manifestations are neurocognitive in nature. And there's a lot of evidence for dysautonomia or general nervous system dysfunction. What we don't understand very well yet is whether all of these have to occur in order for a patient to experience symptoms, or if these are different manifestations that lead to different symptoms that can all be categorized as post-acute infection syndromes. So we set out to study this once we learned of the first long COVID cases at Penn Medicine. And we teamed up with a number of experts at Penn Medicine, including Ben Abramoff, who is leading the long COVID clinic, as well as Sarah Cherry, who is a virologist here at Penn, as well as Mayan Levy, whose PhD student Andy Wong has led this study that I will describe to you now. And so what Andy did early on during the pandemic was to essentially follow a large cohort of individuals that developed these long-term symptoms, that developed symptoms of long COVID or PASC, which is the medical term for post-acute sequelae of COVID-19. And then what Andy did was to look at their symptoms, basically their clinical charts, and to cluster them according to their symptoms. And then she selected individuals from each of these clusters, and she asked them to come back for a blood draw. And what this allowed us to do is to compare individuals with long COVID to individuals with acute COVID-19, as well as those that have recovered to a symptom-free state. And we can then look at their metabolite abundances in the peripheral blood, and you can see that there was a number of molecules that were depleted during acute COVID-19, recovered to normal levels in those individuals that didn't have long-term symptoms, but stayed reduced in individuals with long COVID. And among the most striking association we found was actually serotonin, which you can see here is reduced during acute COVID. This is something we knew from many papers early on during the pandemic. Many groups have seen this. And then in recovered individuals, levels go back to normal, but they stay low in the long COVID group. So that was an interesting observation because, of course, serotonin has many different biological functions, both in the periphery as well as in the brain. So we wanted to know whether this phenomenon is unique to SARS-CoV-2 because, as I described earlier, SARS-CoV-2 is one of the pathogens that can lead to these long-term outcomes, but it's by no means the only one. So we looked at the individuals infected with other viruses systemically, We looked at a range of different viruses that can cause systemic viremia. And in each of these cases, we saw exactly the same thing, that serotonin levels were reduced in these individuals. And that's not only the case in humans, but also in animals that we infect with either SARS-CoV-2 or BSV or a chronic LCMV infection. No matter the virus that we looked at, we always see reduced serotonin in those animals that have persistent virus. For example, here with clone 13, which is a chronic strain of LCMV, while the acutely cleared strain, Armstrong, actually didn't reduce serotonin. And the reason why this is interesting and important to highlight is because there is this theory in long COVID research that much of the long COVID symptoms might be caused by viral persistence. And we actually see evidence in our own cohort as well. This is just a subset of individuals where we looked at viral load in the stool of these individuals. And you can see that a few individuals with long COVID have detectable viral RNA in their stool. So all of this is suggestive of a persistent viral signal that may lead to serotonin depletion. But now, of course, now that we had these comparisons, the question was, how does viral infection lower serotonin levels, especially because it doesn't seem to be unique to SARS-CoV-2 and can be observed in other viruses as well. So we reasoned that one signal that is very common across all these different viral infections is, of course, an elevated interferon response. And the reason why this was interesting is because there is evidence from humans with long COVID that some of these people might have prolonged interferon responses, and that they basically don't manage to terminate their interferon response. And so as we expect, if we infect animals with VSV, we get the induction of interferon-induced genes, exactly the same with SARS-CoV-2 and with chronic LCMV as well. So we wondered whether that's all we need to reduce serotonin levels, basically, if the elevated interferon response is what suppresses serotonin levels. And so we asked this directly by not using a viral infection, but by inducing interferon response, even in the absence of a viral infection, using poly-IC, which, as you know, is a viral RNA mimetic that triggers TLR3. And as expected, we get the induction of interferon-induced genes very robustly using poly-IC. And now, interestingly, poly-IC is enough to reduce serotonin levels, just like those viral infections, which suggests that essentially poly-IC and TLR3 signaling is what leads to the interferon response, and then downstream of the interferon response, serotonin levels are suppressed. We tested this more directly by looking at the canonical interferon pathway. So if we block the interferon receptor, or we knock out TLR3, or we knock out STAT1, which is the transcription factor downstream of the interferon receptor, now we protect the animals that we inject with poly-IC. We protect them from the loss of serotonin in the periphery, which suggests that it's the canonical pathway of viral RNA recognition, TLR signaling, interferon signaling, and STAT1, which suppresses serotonin signaling. But the big question is, how does this happen? What is this question mark here, and how does it lead to serotonin reduction? So we set out to investigate this, and we actually identified multiple different mechanisms by which interferon signaling may lead to reduced serotonin in the periphery. That's what I'll show you in the next few experiments. So we first basically looked at what we know about this pathway upstream. I already mentioned that persistent viral infection, or the presence of viral RNA, will lead to immune recognition and interferon secretion. But now, what do we know about the regulation of serotonin levels? So serotonin, as you know, is derived from dietary tryptophan, which is an essential amino acid. So we absorb it through the gastrointestinal tract, specifically through an apical transport mechanism, which involves two critical proteins. One of them is SLC6A19, which encodes for the amino acid transporter BOAT1. And then interestingly, ACE2, which, as you know, is the SARS-CoV-2 receptor, has basically a second life in the gastrointestinal tract as the chaperone for BOAT1. So these two proteins are needed in order to import dietary tryptophan. And then tryptophan is used as a substrate by enterochromaffin cells, which then produce serotonin. And once it's produced, serotonin is stored in platelets. The large majority of serotonin is in platelets, which is then basically used to transport serotonin in the systemic circulation. And now we found a few different mechanisms by which interferons can basically block this uptake system, as well as the storage in platelets. And I'll show you a few examples of this. So on the one hand, we see that interferon signaling down-regulates the machinery that is required for serotonin, for tryptophan uptake and serotonin production in the GI tract. So you can see here ACE2 is reduced in poly-IC treated animals, SLC6A19 is reduced. As a result, tryptophan levels are reduced, so the precursor for serotonin is lacking. And now we can use a chemical trick to determine whether this transport mechanism is involved, because what we can do, we can use a strategy that was developed around 15 years ago. We can generate a glycyl tryptophan dipeptide, which no longer requires ACE2 and BOD1, but can be taken up through a dipeptide transporter. And when we do this, now we can essentially rescue the tryptophan levels, even though we treat these animals with poly-IC. So this told us that the uptake mechanism involving ACE2 and BOD1, basically the apical transport mechanism for tryptophan, is what's needed and what is downregulated by this interferon response. And now on the other hand, we also found evidence that the chronic interferon response is involved in the storage of serotonin in the systemic circulation, because we found that this leads to hypercoagulation. So you can see here that platelet activity, which we use, which we read out by measuring the levels of CD62P as an activation marker for platelets. So platelet activation is enhanced by poly-IC and the interferon response. And as a result, we get more aggregation of platelets, which you can see here in this flow cytometry assay. Whenever we have platelets aggregating, we'll find them here in this quadrant. And poly-IC treatment definitely leads to more aggregation. And as a result of this, we detect thrombocytopenia in these animals because the platelet count overall after this hypercoagulability events is reduced in these animals. So both of these findings together suggest that serotonin levels are reduced by at least two different ways downstream of this chronic viral signaling. On the one hand, we block the uptake because the uptake machinery is reduced, and so we have reduced uptake of tryptophan, and the substrate for serotonin production is lacking. And then in addition, we have this effect on platelets, which results in essentially the carrying capacity of the systemic circulation for serotonin to be reduced. But now, of course, the ultimate question I want to ask is, why do we care about any of this? Why are we so interested in understanding the regulation of serotonin levels in the context of post-viral infection syndromes? And one of the reasons is what I already mentioned. Serotonin is a neurotransmitter that has many different functions. But what's very interesting is that serotonin may explain, or differential serotonin levels may explain some of the manifestations of long COVID. So we went back to our long COVID cohort, and when these individuals came in for a blood draw, we asked them to fill out a symptom questionnaire. And very consistent with the literature, we find that the big majority of individuals report neurocognitive manifestations. They report fatigue, cognitive difficulties, sleeping problems, anxiety, memory problems, and so on. And while some of these are difficult to model in animals, we can actually address, for example, the memory problems very well, because there are, for many decades, there have been established models of how you can assess memory in animals, for example, using a very simple novel object recognition task, where we expose the animals to different objects. They can familiarize themselves with these objects, and then we come back an hour later or 24 hours later, and we swap out one of these objects to a new one. And then, because the mouse is inherently curious, it's supposed to explore primarily the new object, as long as it remembers what the new and the old object were. And so we can do this test in the context of our poly-IC model or in the context of different viral infections. And whichever model we use, what we see is that a naive mouse, an untreated mouse, remembers the object pretty well and explores with about 70% preference the new object. But now if we cause viral inflammation, for example, with poly-IC or an infection, this memory is impaired. And now we're much closer to the random line of 50% here, where the animals don't remember the object at all. And as expected, this is TLR3 dependent. So if we knock out TLR3, now there's no difference anymore between the poly-IC treated animals and the control. And the same is true if we knock out the interferon receptors. As expected, no effect of poly-IC anymore. But where it got really interesting was when we used interventions with the serotonin pathway that I just described to you. for example, fluoxetine, which is an SSRI, so it enhances serotonin signaling. And now again, we can rescue the animals from this memory deficit induced by viral inflammation. And if we use our glycine tryptophan dipeptide, which, as you remember from a few slides ago, is the one that can bypass the transport system, which is impaired by chronic viral signaling. Now again, we can restore tryptophan levels, we can restore serotonin levels, and importantly, we can restore this memory phenotype, even in the presence of ongoing viral inflammation. So this suggested that this pathway might be involved in some of the neurocognitive manifestation. But of course, the big question was, how does the signal get to the brain? Because I should mention that we actually did not find brain serotonin levels to be influenced by poly-IC. So you may know that the brain pool of serotonin and the peripheral pool of serotonin are differentially regulated. The big majority of serotonin in the body is produced peripherally in these enterochromaffin cells that I mentioned. But the brain can synthesize serotonin as well. And actually, you can see here in this graph that viral inflammation does not impair serotonin levels in the brain. So the question is, how does the brain know about serotonin levels in the periphery? And we hypothesized that one way to convey this signal to the brain is through sensory neurons like the vagus nerve, because these sensory neurons are very rich in serotonin receptors. The vagus nerve specifically expresses 5-HT3 very highly. And then we hypothesized that through the vagus nerve, the signal is sent to the brain, and then through a polysynaptic pathway may influence the hippocampus and other areas of the brain involved in memory formation. We had first evidence from this by looking at the nucleus tractus solitariae, which is, or NTS, which is the area of the brainstem where the vagal terminal synapse. And you can see here that poly-IC treatment reduces the activity of these brainstem neurons in response to a novel object. But more importantly, when we come back with our memory test, and you can see here the control is impaired as you would expect from the previous slide. And if we supplement 5-HTP with this, which is the biosynthetic precursor of serotonin, now we can rescue the animals, and we can even rescue the animals with non-specific activation of sensory neurons using capsaicin. So as you will know, capsaicin is a ligand for TRPV1, and TRPV1 is highly expressed on sensory afferents, including sensory afferents of the vagus nerve. And if we just stimulate these sensory neurons using capsaicin, we can now essentially bypass the effect of viral inflammation, and we can completely restore cognitive abilities in these animals. And I think what's even more important is that we can achieve the same effect using a pharmacological 5-HT3 agonist in the periphery. So now if we essentially recapitulate serotonin signaling just by stimulating these 5-HT3 receptors in the periphery, again, we can restore memory capacity, even though we treat these animals with poly-IC. So all of this collectively suggested to us that there's this upstream pathway of chronic viral recognition, chronic induction of interferons. And then downstream to this, there are at least these two mechanisms that I discussed in terms of blocking tryptophan absorption, reduced serotonin production, and reduced carrying capacity for serotonin in the systemic circulation because of platelet dysfunction. And as a result of all of this, we have reduced activity, reduced triggering of serotonin signaling through 5-HT3 in the peripheral nervous system, including the vagus nerve, which then leads to these neurocognitive sequelae that we can observe in the animal model. How much of this pathway is true in humans is something we still don't know, but we're actively investigating. And on a larger scheme, I think this brings us back to where we started, because I've shown you a few examples in the context of inflammatory bowel disease, as well as these post-acute infection syndromes, where we may be able to add a third component, a third commonality that is shared by many of these very common conditions, which is what we call defective interoception or defective communication between the body and the brain in general. And this is something that may not be unique to the stressful life event that leads to exacerbated peripheral inflammation, and may not be unique to post-viral conditions, but could potentially be extended to many of these other conditions that we looked at on the first slide, because there's growing evidence that defective interoception might be a shared feature of many of these conditions. So I think I'll stop here, but I will not finish without mentioning many of the people that contributed to all of this work. I mentioned Nicholas Blank, a member of the lab who is very interested in the connection between stress and inflammatory disease. as well as 2 alumni of the lab, Marcus Schneider and Yelim Alvarez. And I mentioned our collaborations with the labs of Mayan Levy, Ben Abramov, Sarah Cherry, as well as Bob Hoykeroth, who is our main collaborator on everything that involves the enteric nervous system. I also want to point out a lot of support by various people with obtaining clinical samples and collaborating on human cohort studies, as well as many other people, both at the University of Pennsylvania and beyond, that have supported us and continue to support us with many different aspects of these studies. So with this, I'll finish. Thanks again for inviting me to be here today. Thanks to my kids who managed to continue sleeping through the talk. And I'll be happy to take any questions. Thank you.